Very good evening to all the NEET aspirants, NEET 2026. This is your Prasanna Ma'am from NEET World. We are here to discuss the last important segment of the chapter, that is centrioles, cilia, flagella and cytoskeleton. Okay. So, first let us discuss cytoskeleton in detail, then after we will go into the details of centriole, cilia and flagella. Cytoskeleton, where do you observe this? Okay. Cytoskeleton you observe both in the plant cell and animal cell beta. Okay, you observe this both in the plant cell and animal cell. What is cytoskeleton? The name itself is saying that this gives shape to the cell and this maintains the cell structure and cell shape. As such, cytoskeleton will be having the filaments. What are composed in the cytoskeleton? Cytoskeleton is composed of intermediary filaments, microtubules and microfilaments. Microtubules, microfilaments and intermediary filaments. So, you tell me what is cytoskeleton composed of? First is microtubules, microfilaments, intermediary filaments. From these three, what we have to learn, what is more important? I will tell you, see. You have to look into what are the proteins involved in microtubules, microfilaments and intermediary filaments. What are the functions with respect to microtubules, microfilaments and intermediary filaments. For microtubules, if you look into better, microtubules, these are unbranched hollow protein structures. Okay. These are unbranched hollow tubules made up of the protein tubulin. Microtubules are made up of what protein? Tubulin. Remember that. When it comes to the functions, microtubules are primarily involved in spindle fiber formation. Spindle fibers, those which attach to the kinetochores, and they will help in what? Centromere splitting during metaphase, anaphase. So, that spindle fibers are formed with the help of what? Microtubules. Second is what? Chromosome fibers. Okay, chromosome fibers and basal bodies. You can see basal body in the flagella. That basal body is composed or it is attached to the cell membrane and cell wall through what? Through microtubule and cilia and flagella. In cilia and flagella also you can see the microtubule arrangement in cartwheel shape that we are going to see now. Okay. So, all the spindle fibers, chromosome fibers, basal bodies, cilia and flagella are composed of what, Nana? Microtubules. Clear with this now? Second, this functions as cytoskeleton that gives proper shape to the cell and maintains. Third is for intracellular transport. After Golgi bodies which are releasing out the vesicles, Golgi bodies will release out the vesicles both for intracellular targets and intercellular targets. For the reach out of that intracellular targets, what structures are helpful? Microtubules. So, for intracellular transport. Next is control of shape of wall-less cells and nucleus, to maintain the shape of wall-less cells as well as the nucleus shape. And this helps in movement of nuclei during cell division. As it is helping in the spindle apparatus formation, they will help in the movement of nuclei during cell division as well. They help in the formation of cell plate in the plant cell. Okay. From cell plate only the first formed cell wall appears, that is middle lamella. It comes from what? Cell plate itself, and that cell plate is formed from what? Microtubules. Next is what, Nana? Cell movement. Cell movement all across that environment is modulated by what? Microtubules itself. Fine. Cellular movement is required, like RBCs will be moving all through the circulatory system. As such, cellular movement is modulated by microtubules. All this set of functions should be in your mind for sure. Okay. And what is the protein composition of microtubules? Tubulin protein. Clear with microtubules now? Now, look into the microfilaments. These are narrow cylindrical rods. They are made up of protein filaments, actin. Okay. You can include actin and myosin as well. So, what are the main functions of microfilaments? First and foremost function is cytoplasmic streaming, and it gives support to the cell. Next, in formation of myofibrils, which are also called as muscle fibrils, and these are helpful in the formation and movement of microvilli in the small intestine, and formation and retraction of pseudopodia in amoeboid protozoans like entamoeba or amoeba. Okay. So, pseudopodia is also called as what? in simpler ways? False feet, right? For the movement of pseudopodia, what is required? Microfilaments are required, and this microfilaments are utilized for endocytosis and exocytosis. Here I would like to tell you two things. Endocytosis is of two types, Nana. You can call pinocytosis. Yeah, pinocytosis is also called as no. Pinocytosis is also called as cell drinking. For example, for suppose you observe like this is a cell. There is some particle, lipid droplets, liquid particles around the cell membrane. Taking up those liquid particles into the cell is what? Cell drinking. That is called as what? Pinocytosis. This is cell drinking. Next, phagocytosis, engulfing food particles around the cell membrane. Those food particles engulfing is called as what? Cell eating. Phagocytosis, that is cell eating. Yes, that is cell eating. Remember, endocytosis is of two types, phagocytosis and pinocytosis. Pinocytosis is cell drinking, phagocytosis is cell eating. Thank you. I don't know who's this, the everything zone. Thank you for the compliment. Next, exocytosis. Okay, exocytosis is also called as cell vomiting or effaggy. Whichever cellular constituents which are not required for the cell, those are egested out. That is also called as what, Nana? Exocytosis is also called as effaggy or cell vomiting. Cell vomiting. Clear right now, Kunal? Right, sir? Next, formation of spindle apparatus. Don't get confused. This is with respect to microtubule constitutes spindle fibers, but microfilaments will help in formation of spindle apparatus. Spindle apparatus includes what? Spindle fibers and asters. Yes, asters and spindle fibers together called as spindle apparatus. They help in formation of this. Whereas microtubule constitutes spindle fibers. Don't get confused. Next, movement of cell components. Okay. Cell components moving inside the cell is for microfilaments. One solid thing, keep it in your mind, for intracellular and intercellular transport, microtubules are helpful. Microfilaments, strictly it is for cytoplasmic streaming and intracellular targets only. Clear, right, beta, with microtubules and microfilaments. Coming to intermediate filaments. Intermediate filaments are solid, unbranched filaments. They are formed of variety of proteins, varying of many proteins. As such, the functions of intermediary filaments include, first we have to know where are intermediate filaments present. They are present inside the nucleus. And what is the major protein composition inside for the intermediate filaments? Lamin proteins. Lamin proteins. Okay, ma. So, what are the functions involved in this? Formation of nuclear matrix, okay, inside the nucleus, formation of nuclear matrix, and support of membranes, supporting array of cytoplasm, supports contractile units, and this will maintain mechanical strength of nervous tissue. So, such unique functions are performed by intermediate filaments. It is very rare that you will get question from intermediate filaments, majorly focus on microtubules and microfilaments. Okay, beta? Next. So, now you have understood what are microtubules. Let us get into the centriole. First you tell me, where do you observe centriole exactly?
Poles of the cell.
Okay, which type of cell? Which type of? Yes. So, centriole strictly belongs to what? Animal cell. Centriole strictly belongs to animal cell. So, if I have to consider this as a cell beta, okay, inside a animal cell, inside the centrosome, don't get confused between two terms, the centriole and centrosome and centromere. Centromere is primary constriction of the chromosome. Centrosome is constituting two centrioles. How centrioles are arranged inside the centrosome? Inside centrosome, centrioles are arranged as two perpendicular structures like this. Like this, two centrioles are arranged. Okay, two centrioles are arranged perpendicular to each other. inside the centrosome. These are non-membrane bound organelles inside the animal cell, okay, only unique to the animal cell. What is the main function of the centrioles? Formation of spindle fibers, spindle fibers, or you can simply say as spindle apparatus. Just a second. Spindle apparatus. The spindle apparatus will include what, Nana? Spindle fibers, very good, and osteoids. Fine. So, this is ultimately helpful in what? Cell division, right? Cell division of animal cell. Then how cell division of a plant cell is modulated without centrioles?
Cell membrane of the plant.
No. How cell division is processing inside a plant cell without centrioles? In animal cell, centrioles main function is what? Cell division. How they are helpful in the formation of spindle apparatus. In plant cell, then how spindle apparatus are formed? No, you are almost there. If I consider this as a plant cell, in plant cell there is no centrioles, but then there is something called as microtubular organizing complex. Okay? This microtubular organizing complex will help in spindle apparatus formation. Okay? So, what is this? Microtubular, yes, microtubular organizing complex. Fine, this is microtubular organizing complex. Now, let us look into the cartwheel shape of the centriole. We got to know where centrioles are present, what is the function of the centriole, in what they are included inside the centrosome. In plant cell, how the function is compensated without the centriole. Now, let us look into the clear structure of the centriole. So, this centriole beta, if you see, at the center there is a proteinaceous hub. There is no microtubule at the center. What do you call it as? Proteinaceous hub. From this proteinaceous hub, there are 9 radial spokes that are radiating out. How many radial spokes? 9 radial spokes. To this 9 radial spokes, for each radial spoke, there are 3 microtubules. How many microtubules? 3 microtubules for each radial spoke. Fine. As such, let us name these 3 microtubules A, B and C. C of one microtubule is connected to A, CA connections we call it as. Fine. So, as such, total how many microtubules will be there? Center one proteinaceous hub, and to that 9 radial spokes are there. To each radial spoke 3 are present. So, it is very simple, right? 9 into 3, 9 into 3, total 27 microtubules. Clear? This is with respect to centrioles. Everything is clear with respect to centriole, right? Fine. Now, we will look into the cilia and flagella also, and then we will see where centrioles are again present, how they are helpful in flagella structure formation also. For cilia and flagella, you observe, cilia and flagella. Unlike the centrioles, in centrioles though, at the center you have proteinaceous hub, but here you have 2 central microtubules. Okay? Central microtubules, these are microtubules. How did you get this structure? Where the flagella is present? For let us suppose this is the bacterial cell wall. Yeah.
Protruding out.
Yeah. Yes. So, flagella will be the extensions of cell wall, right? So, it will be extending like this. Fine. So, flagella will be having 3 parts, right, Nana? What are they? Basal body, hook and filament. So, you have basal body, hook and filament for the flagella. This basal body is connected to the cell wall with the help of centriole. Remember this point. Fine. Now, this core region at the center, listen first beta, this core region at the center, you call this as what? Axoneme. Core region at the center, what do you call it as? Axoneme. When you transverse sect this axoneme and see, you can see this microtubular arrangement. As such, center you have two central microtubules. These central microtubules are enclosed by a central sheath. Fine. From this central sheath, you have 9 radial spokes. Each radial spoke will be having how many microtubules? 2 microtubules. Fine, ma? So, here central microtubules are there. Covering this you have central sheath. From this central sheath you have how many radial spokes? 9 radial spokes. To each radial spoke 2 microtubules are present. On total, how many microtubules are present in cilia and flagella?
20.
Cilia and flagella, total how many microtubules will be present? 2 central plus 9 radial spokes, each radial spoke is having 2. So, 2 plus 18 will be 20 microtubules. Clear right now? This is about cilia and flagella. Now, let me put simple question. Okay, I have already told you that inside the centrosome, you will be having 2 centrioles which are perpendicularly arranged. One centriole is having how many microtubules? 27 microtubules. One centrosome will be having how many microtubules?
54.
Very good. It is 54 microtubules. Clear, right? Easy, right? Enough. You tell me, what are the motile structures in a prokaryotic cell?
Cilia
Only cilia?
and flagella.
No. For a prokaryotic cell, no. The only motile structure is flagella. Yes. And there are 2 non-motile structures, you call it as pili and fimbriae. We have already discussed, right? As such, flagella will be having 9 plus 0 arrangement like centrioles in a prokaryotic flagella. And what is the protein content that is present here? Flagellin protein. Yes, flagellin protein. Okay, flagella with 9 plus 0 microtubular arrangement, and it has flagellin protein inside the prokaryotic cell. When you have to look into the eukaryotic flagella, here you see, ma, the flagella is having 9 plus 2 arrangement. Flagella is having 9 plus 2 arrangement, and the protein present inside the flagella is tubulin protein. And here not all the parts of flagella will be having same 9 plus 2 arrangement. Okay, I told you one important point, that the basal body is connected to the cell wall through what? Centriole. So, for basal body the microtubular arrangement will be 9 plus 0 only, ma. For basal body the microtubular arrangement will be 9 plus 0. For hook and filament it is 9 plus 2 arrangement. Okay. So, this cartwheel, if you can imagine how this is moving, like this. So, how flagella will be moving? In this way only. How centrioles are moving towards the opposite poles? Through this movement itself, because cartwheel moves like this. This is what the movement of microtubules is happening. Clear, right? So, now you are clear with cytoskeleton, you are clear with centrioles. Okay. What will be the centriole arrangement and how they are present? Yes, inside the centrosome, and how they are helpful for formation of spindle apparatus. In plant cell, how is that function compensated? By microtubular organizing complex. What are the major differences between centriole, cilia and flagella? Here in the centriole, at the center you will have what? Proteinaceous hub, where in cilia and flagella you have 2 central microtubules. Okay? And to the 9 radial spokes in centriole, to each radial spoke you have 3 microtubules. Cilia and flagella will have for each radial spoke 2 microtubules. What are the total number of microtubules in a centriole? 27 microtubules. What will be the total number of microtubules in the cilia and flagella? 20 microtubules. Once again you just recall all the, this thing. In a prokaryotic cell, what are the only motile structures? Flagella. What will be the microtubule arrangement here? 9 plus 0. And what will be the protein composition for the flagella? Flagellin protein. Here in a eukaryotic cell, if you take 9 plus 2 tubulin protein, and it has basal body, hook and filament. Basal body
is connected to the cell by centriole.
So it has 9 plus 0 arrangement, hook and filament is 9 plus 2. Once you just recall for 1 minute, have a glance on it. Online students, you can take a screenshot of it. Look into all the functions in a more detailed way. Fine? So yes, with this we are done with what? Cytoskeleton, centrioles, cilia and flagella. Fine, in cytoskeleton we are dealt in detail about microfilaments, intermediate filaments and microtubules. With this we end the chapter cell, the unit of life. In our next session, let us exercise cell cycle and cell division, major concepts from that. Thank you.